Very good afternoon. So now today we discuss the seed structure and seed dormancy. So this one is very important topic: structure and dormancy of the seed. So in any seed, so yesterday we discussed that one, but seed is the mature embryo after fertilization. Yesterday we discussed that one seed growth and discuss. So now the any seed is consisting some following parts are there: seed coat, embryo, these are there. So what is seed coat? It's the protective coat made up to two layers. So outer thicker layer is the testa and head. Inner one is the tech. The present as an embryo, which is enveloped to protect the embryo and enters first answer. So these the seed coat which have to protect the embryo enters the from that desiccation and some mechanical effect of environmental condition. Sometimes it is protected from the other damages, means due to the some pathogens, means microorganisms or insects also. And the seed coat also sometimes which helps to the seed dispersal also. Seed dispersal. And another one is the embryo. The embryo, it is an so rudimentary plants present in axis found with one tip known as the plumule. The plumule is responsible from the shoot portion, and other tip that one we call as the radicle. That's the plumule. The radicle are also there. That radicle to form the roots affects from radicals. The portion of embryonic axis extends above the cotyledon, which is known as the epicotyle, and below the cotyledon, it's called as the hypocotyle. So this is the structure of seeds structure already I shown to you the picture. This is the seed structure, monotype seed and diploid. So next one is the storage of food. Storage of food in the form of endosperms only. Endosperm is a thick and massive structure which is made up of the elongated cells consisting the abundant starch grains are present. On the basis of the presence or absence of endosperm, so seeds are categorized into two types of so albuminous and ex-albuminous seeds. Means the endosperm is present, the seeds called as the albuminous seeds. Ex-albuminous seeds, the endosperm is absent. That one we call as ex-albuminous seeds. So here in the monocots, all the monocots are albuminous seeds only. That is the example, wheat, rice, and other cereals. So dicots, but all the dicots are the ex-albuminous seeds, except castor. The next topic is next part is the cotyledon. So what is the cotyledons are there? It is an extension of embryo on the basis of number of cotyledons. The craft may be divided into two types are there, that is monopod and dipod. You know that. So monocots means which is present only one cotyledon, whereas the other dicotyledons, three cotyledons are there. But in gymnosperms here, 2 to 18 cotyledons also present. And in the monocots, the scutellum situated between the endosperm and embryo is the single cotyledon. That is all. So the food structure known as the seed, that is this seed, some of the following parts are there. So there is the caryopsis. Following single-seeded fruits are usually considered as the seed. There are the caryopsis, akin, cytoplasm, and nuts. So what is caryopsis? That means single-seeded fruit that will be called the caryopsis with the fused the fruit and seed wall. Fruit and seed wall both are the fused formed as a caryopsis. Example, wheat and the sorghum. That is the caryopsis. So next one is the akin. Here, akin is also single-seeded fruit where the pericarp can be removed from the mature seed. That is the sunflower, example, sunflower and safflower also. Another type of fruit here is schizocarp. So this schizocarp is a dry fruit which is separated into two or more units at maturity stage. That is example as coriander, sometimes carrot. And another type of fruit here, nuts. So nuts fruit with a thick and hard shell-like fruit ball in there. Examples are these are the some types of the fruits are the single fruits. Okay. And so structure of crop seeded. are there. The monocot seeds are crafted from the granule. So these monocot seeded crops, so there is example wheat, rice, and sorghum, maize, all. These all are the monocot seeded crops. So these are the carry-up, all are the carry-up. The endosperm of these seeds fed upon non-living cells bearing that one is hyaline layer. Out of the layer is hyaline layer. So a very small embryo made up of shell-shaped single coordinated scutellum. And a short axis lays and your root at one end of the endosperm. The epithelium layer separated the embryo and endosperm. The cotyledon forms a protective sheath known as the epipotyle on the pleural and polarized on the rad. So this is the epipotyle and polarized. Epipotyle to protect the pleural and the polarizer to protect the radicle. So another one is the wheat. Another seeded crop that is the wheat is there has some at the dorsal and wheel at the ventral side. The embryonic end of seed is known as the coral end, whereas other end is known as the brush. Brown in color. Another one is the rice, another type of seed. It is the caryopsis. So tightly closed in the ligum and scalia. The endosperm of rice of two types are there. That is the malia, which is highly breakage during the milling, and vitreous. So less breakage and non-sticky. Two types of rices. So next, so next one is the sorghum. Sorghum seed. So sorghum is the pericarp part. This one is the pericarp. So pericarp, which is consists of an epicarp, mesocarp, and endocarp. The hilum turns black at physiological maturity. The endosperm consists of a layer of alluvium cell and an outer carneous endosperm surrounding the central flori or starchy endosperm. Next one is the maize. Maize, the seed is conical in shape with a broad and round upper end and sharply pointed at lower and embedded in the cope. White and oval area containing the ridge-like structure, which is indicate the portion of embryo. The yellow part of the grain, which is called as endosperm. And other seeds also there, onion seed. It is an monocotyledon hexalbuminous seed. Embryo is spiral twisted and cylindrical in shape. So most of the potash embryo is made up of the cotyledon. And some other dicot seed crops are there from leguminaceae families. So dicot seed of crops, these are the hexalbuminous dicotyledon seeds. The embryo is made up of embryonic axis and two fleshy cotyledons. The seed is round to oval, spread by protective seed pod, the scara, phylum, and microphyle. The radical portion of embryonic axis projects behind the cotyledon. The other end of the embryonic axis is the plumule present in between both of the cotyledons. In some species, gut-like outgrowth is present below the phylum called Sophiolus. Sophiolus. So other achenes, that is already discussed that from sunflower and starflower. These are the dicot single-seeded fruit covered with the fruit wall having some ridges. Usually, a very thin layer of endosperm is present in between outer covering of the seed and cotyledon. The cotyledons are flat with the clear nerve. The embryonic axis is a short and a radical is present in the extended from at the one side of. Another type of seed is castor. So it is a large seed covered with a hard seed coat. At one of the end of the seed, parenchyma is present as a whitish outgrowth of integument. The hilum is present at the base of and partly hidden due to the parenchyma, whereas the micropyle is located between the parenchyma and the hilum. The testa has white and brown spots on the removal of the testa. A white, oblong, flatted endosperm covered with a thin segment layer became visible. Embryo is embedded in the endosperm. These two cotyledons are large and flat with a clear vein. The plumule bears a number of minute leaves near the apex, which remains hidden between the two cotyledons. So system is a flattish, ovate, white to light brown small seed with a dark base, front margin line, and an equally pale central line on the one face. Seed is hexagonous with a straight embryo. So linseed is the flat to elliptical, ovate brown, and glassy seed. Another one example. Dark reddish brown to brownish black seeds. A scare is visible on the seed coat. So lettuce. The seed with the both endosperm and cartilage are store the food. Another type of seed food, the beetroot means beta vulgare. The seed with the endosperm, cotyledon, and nucleus tissues are store of the food. So another one is the coffee arabica. So nucellus and store the liquid and and open with smooth surface. Seed coat is thick, two parts is comprised with seed. This is the seed structure of seed. Next, so seed dormancy. So seed dormancy can be defined as the state or condition in which the seeds are prevented from germination, even under the favorable environmental condition also. The favorable condition, that is the type of dormancy. Here, two types of seed dormancies are there: exogenous seed dormancy and endogenous seed dormancy. Any problem? Fourth తర్వాత ఎండ ఉన్నది కదా. Exogenous seed dormancy, that is the physical dormancy and mechanical dormancy. The seed coats are impermeable to water. The seeds are at a low moisture level. The legumes are an example here. In the dormancy, and it's a layer which becomes impermeable to water. Clearance cells in the mucilaginous other cell layer are 100 that is the hardened endocarp or the three regions that seed coats became impermeable to water. So the seed coats developed during the last stages of seed development. With the area show that as the depth of puncture to the seed coat increased, so did the permeability of the seed coat. Mechanical dormancy. The seed coats are too hard to allow the embryo to expand during the germination. So causes include structure of seed coats or remaining fruit occur in olive. In nature, The coats are softened by environmental agents such as acid in guts, microorganisms in warm environmental forest fires. To overcome horticulturally scarified with the sandpaper or sometimes hot water, acid, warm moisture, environmental fire. These are the use the immature fruits. Once scarified water can be get in, have to carefully because the seed is also more susceptible to damage by the microorganisms. So through the chemical here, the presence of chemical inhibitors in the outer covering of many fruits and seeds. This occur in the fleshy fruit hulls and capsules of many dry fruits. Example: apple, citrus, grapes. Very acid is dry. This kind of may also be present in tissues, the embryos such as in the sperm. And the oxygen may be restricted by mucilaginous layer occur in spinach of some asteraceae. So in nature, the overcome by heavy rain, which can leach the inhibitors in tropics, deserts, some salts as ammonia. Or change water daily for several days, chilling of a few days, existing of embryo, use of hormones, especially gibberellic acid. Endogenous in what is the endogenous dormancy here? The morphology, the embryo of the need additional embryo growth after the seed is separated from the plant. Described in the terms of how much space embryo takes up in the seed. In several herbaceous flower seeds such as um, ranunculus, poppy, woody species, are combined kind of means hard seed coat. So rudimentary about the pre-embryo stage may be inhibitor present. Ranunculaceae, Paperaceae, Araliaceae, these are there. To overcome artificially alternate warm and cool temperature, also hormones such as gibberellic acid export to cool temperature. Some tropical species require extend period at high uh, temperature for fully developed development of embryo, where date plums often takes place the several years to germinate. So takes this long time to reach full embryo development. So often high temperature will accelerate these dormancy. Dormancy breaking methods. So seed dormancy breaking methods of clarification and stratification. First one is the physical dormancy, that is the scarification.
Any treatment may be physical or chemical that weaks or softens seed coat is known as a scarification. So the scarification through the acid means mechanical or physical treatment or sometimes biological treatment also. Acid scarification by using the concentrated H2SO4. So concentrated H2SO4 in 100 ml per kg of seeds. For the 2 to 3 minutes treatment, dormancy can be overcome in the some seeds. The duration of treatment will vary and it is depend on type of the nature of seed coat. So here tree crops, so 1 to 3 hours, rose seeds treat with the seed partially with the acid and then give with the warm scarification. Mechanical scarification. The seeds are rubbed on the sandpaper or with the help of mechanical scarifier or by the puncturing of the seed coat with help of the needle to enhance and increase the moisture absorption by seeds. Example, bitter gourd.
Another method is done digestion gut is uh, next one is hot water treatment. Hot water treatment it is an effective in case of leguminous tree crops seeds. The seeds should be soaked in the boiled hot water for 1 to 5 minutes. Some crops like Bengal gram and groundnut, hot water treatment for more than 1 minute is found injury to seeds. Next one is the stratification treatment. Stratification when the seed dormancy is due to embryo factor, seed can be subject to stratification treatment. So cold stratification and warm stratification. Incubate the seed at low temperature of means 0 -5 degrees over a mature substratum for 2 to 3 days to several months. It depends on the nature of seed and kind of dormancy. For example, cherries and oil palm seeds. Warm stratification. So some seeds is required temperature around 40 to 50 degrees after 2 days. Example, paddy seeds. In case of oil palm, it is required the temperature of 40 to 50 or 2 months for the breakdown seed dormancy. This one is the leaching of metabolites. So these seeds can be soaked in water for 3 days, but once in 12 hours, fresh water should be changed to avoid the fermentation. The seeds can be soaked in running water for a day to leach out the inhibitors. So, example, coriander or sunflower. So, temperature treatment. Here, low temperature treatment and high temperature treatment also there. So, plants which grow the temperature and cooler climate, which require the period of chilling after breakage of dormancy. High temperature treatment. Normally, high temperature treatments are exhibited by early flowering winter animals. So, alternate treatment. alternate temperature change. Most of the plant species which grow in the temperate and cool temperate regions is required alternate temperature for breakdown of the seed organs. So another one is the light fire treatment. Many shrubs and trees, tropical and semi-arid regions have extremely hot seed pod, in which is the seed pod is very aggressive water. The dormancy in such seeds is clearly seen in frozen and may be breakdown by exposure to extreme heat, such as the fire. So, for example, Saluna vulgaris, that is the seed dormancy is breakdown by the fire only. Okay. This is the seed structure and seed dormancy. So next one is the seed storage. Seed storage, there is in the banks, seed banks are there, or other gene banks. So seed gene banks are set with the objective to minimize the loss of viability and genetic integrity of seed material during the storage and regeneration process. The gene banks means seed gene banks are for orthodox species can be dried to very low seed moisture. These are three types of seed bank storage, that is the short term, medium term, and long term storages. So short term storage means short term storage of seed banks. These are generally maintained at the temperature of 15 to 20 degrees, and RH means relative humidity here 30 to 40%. The breeder material and germplasm material processing for long term can be stored short term storage banks. Another one, the medium term storage. The temperature here will be 5 to 30, and the seed moisture here 6 to 80%, 6 to 8 are adequate for the medium term storage model. It can maintain the seed longevity for a period of 25 to 35 years. These storage modules are meant to maintain active collections, medium-term conservation and regeneration, multiplication, distribution to bona fide users, and evaluation are other important activities. The accession size comprise of as large quantity as possible to avoid the frequent regeneration. So long-term storage module, the storage condition, long-term prefer and temperature here that is -10 to -20 degrees and non-control of relative. Only the basis of collection are maintained in these conditions, and it can maintain the seed anywhere from 50 to 100 years. The base of the collection it is maintained this seed anywhere here 2000 seeds for the self-pollinated crop, and 40,000 means 4,000 seeds for the crop pollinated. Generally prepared for the storage. So around 500 seeds must be stored. These seeds dried moisture contain around 3 to 7%, and these are stored in the hermetically sealed the container. So standard for the seed gene banks are standard here we have to maintain. So seed drying. First one is the seed drying. The seed should be dried period to place the seed containers. Sun drying or shade drying, natural air drying or some hot air drying, and use of this kind the sealed container are generally used. The lower the seed moisture for the prolonging longevity of seed using the dehumidifier, drying the seed around 10 to 20 degrees and 10 to 50% really. So seed cleaning. The seed stored should be free from the weeds, the pest and disease crop scientific and weeds, pests, diseases associated with seeds are preferred. So storage container. The storage container are different for the types of storage purpose. Generally, the paper bags, plastic bags are used in short-term storage. So plastic container and the plastic pouches, the medium storage. Whereas the long-term storage module, the aluminum container, multi-layer pouches, polythene bags here. So monitoring the seed viability. The minimum acceptable limit of germination here is 65 to 85% for gene banks. It will be assessed for the germination test. A minimum of 100 to 200 seeds are like used for germination at the start of seed storage. The period of viability monitoring is 10 years for base collection and 5 years for active collection. The next aspect is the regeneration. Regeneration should be undertaken when the viability falls below the 85%. The seeds of 100 plants or more seeds of provide large classes of alleles. The seeds for regeneration should be initially such as those as possible to the original sample. Information management. Plant genetic resources database is very important at the national and international, regional and global level also for the possible future crop improvement programs. So some of the database important for the management seed banks, that the germplasm passport database. So this data is referring to identify the and history of the accession collection at the time of collection time. Germplasm characterization database. So this germplasm database, so in individual accession plotting of these characters that are highly heritable, which can be seen by the naked eye and are expressed in all of the environmental evaluation. Germplasm evaluation, germplasm evaluation database, and the gene bank conservation database, germplasm exchange Maintain the module. These are the some important. Maintain the modules also. So a voltage stabilizer to protect for the voltage functions built in the electrical control panel with operation information. That's important. Safety and security modules also very important. Safety and security. And another topic is the seed packing. So seed packing involves the place and control, which is the weighed or counted sample of seeds in a container. So which then be hermetically sealed for the subsequent storage. So these seeds packet can be prevented absorption of water from the atmosphere after drying and keep the accessions separate and avoid the mixing others. Prevent the contamination from the insects and diseases. Time of seed packing. So it's very important. But the best time to pack a seed is immediately after moisture content has been determined to the required limits for the safe storage. So dry seeds will be reabsorbed, reabsorbed moisture from one more humid ambient air. So after processing and treating the complete seeds are packed into containers of specified a new way. The packing or bagging is essentially the last operation seeds are handling bulk. The packing consists of following the operations are there. So filling up seeds, bags, extra weight, placing the leaflet in the seed bags regarding improving the cultivation practice, attaching labels, certification stage, certification tags on the seed bags, and sieving of the bags, storage of the seed bags. Among the above two methods, that is oxygen inflated bags with sufficient water is considered good due to their advantages like to reduce the damage to the seeds and easy handling. So further, the seeds are in the batches, the bags are therefore even if mass morality occur that will affect only lesser number of seeds whole lot. So the seeds can be transported in the carrier, either the inflated bags or in oxygen supplied trunks for distance to be determined based on the following means external temperature, number of seeds in a particular bag, size of seeds, metabolic wastages, and the nature of seeds. So possibility for the packing in road. So the one is the types of storage. So types of storage is also there. In this one, types of storage here we are to discuss. Types of storage means storage at the ambient temperature and humidity. Dry storage with the control of moisture content but not temperature. Dry storage with the control both the moisture and content temperature. So dry storage for long-term gene conservation. These are the cryopreservation types of these. Cryopreservation means genetic seeds placed in the liquid nitrogen. That is very important in the cryopreservation. So next one is the storage container. So here we are to storage container. We are moisture, moisture pervious and pervious, moisture resistance. The labeling, another one is the labeling also. Labeling is responsible for the making labeling. This is also very important. Labeling. Okay.